Hello everyone, I am creating this video to explain about the research assignment that has been assigned to you. If you go to your assignment module, you'll see that under research assignment there is a research paper under the title Journal Club. So I'm going to give you instructions on what you're supposed to do with this assignment. So you have been assigned this original research paper from Cell Journal that is entitled Oligopeptide Signaling Through TBGPR89 Drives Trypanosome Quorum Sensing. So I'm going to explain you what is the background of this study. Trypanosoma brucei is a protozoan parasite. So it's a single cellular protist that lives in your blood. So this is a blood smear and these are the parasites that we are talking about. This parasite is mainly prevalent in sub-Saharan Africa, where they have the vector tsetse fly, which transmits this parasite from the infected individual to a healthy individual. So they bite the unhealthy person, suck the blood out of it, and then they bite the next individual who is healthy and put these parasites in their bloodstream. Trypanosoma brucei has an excellent ability to change their surface proteins, because of which our immune system is not able to deal with these parasites, because once your immune system is ready, these parasites change their surface coat and it becomes harder for your immune system to keep recognizing them. People who are infected with these parasites have some milder symptoms or very non-specific symptoms like fever, swollen lymph nodes, pain in joints, but eventually this parasite travels to the brain and there it establishes the neurological symptoms like confusion, sleep disturbance. That's why the disease caused by this parasite is called sleeping sickness. Trypanosoma brucei has much simpler life cycle. In human beings, they are present in the bloodstream. They multiply by binary fission. Eventually, they enter the brain and cause sleeping sickness. They can also infect cattle and cause the wasting of muscles. And that disease is called Nagana. From the host, this parasite is picked up by the vector tsetse fly. In the tsetse fly, the parasite can multiply and eventually enters the salivary glands. And whenever these small little insects bite a human being or a cattle, they can drop these parasites in the bloodstream of the host. But there is an interesting thing to note about parasites. Parasites live in the body of their host. They multiply, that means they reproduce, they grow, and they utilize resources from the host. But they do not want the host to die, because host is their main source of shelter, food, and all other resources. Now, Trypanosoma brucei proliferates very vigorously in the host body. This form of parasite is long and slender in form. But when they reach a certain density in the blood, they do not want to keep multiplying continuously, because that will cause so much damage in the host that the host will die. But Trypanosoma brucei lives in their host for several years, and the host is still alive for several years. So what happens is, this proliferative form of parasite differentiates into a short stumpy form, and this short stumpy form gets arrested in the G1 or G0 phase of their life cycle. And you all are familiar with the different phases of interphase, which is G1 phase, S phase, and G2 phase. So they get arrested in the pre-mitotic phase. Before undergoing mitosis, they get stuck in the life cycle and then they don't proliferate. So for a very long time, it was a question for the scientists to understand what makes these proliferative forms to get differentiated into the short non-proliferative forms. And for a very long time, it was believed that there are some external factors called the Stumpy induction factors SIF, which would lead to this differentiation. So it was as recent as 2019 that the scientists were able to get some clue about what this Stumpy induction factor could be, which in this paper you will see that it's an oligopeptide that leads to the differentiation of trypanosomes from the long slender to the short stumpy forms. So for this journal club, you are expected to give a background of the study and summarize the methods used by the scientists to understand what could be the stumpy induction factor, what are the properties of a factor that will lead to differentiation of a long slender form into a short stumpy form, and then discuss what results they obtained and what were their conclusion, and what is your opinion about the study, or could they have done something differently, or do you find any limitations in this study.